Hey everyone, in this video we will talk about cell cycle. So if you haven't subscribed to my channel, please subscribe it so you will get notified every time when I upload a new video. So starting today's lecture, this is a diagram of chromosome, the part of our nucleus that contain all the genetic information that is passed from generations to another generation. This is the diagram of chromosome. The two chromatids are joined by a centromere at the center. This is the diagram of cell division, in which a parent cell is divided into two daughter cells. This is the separation of chromosomes and then later the duplication of cell. This is the carrier parts of the cell, the organelles that involve in cell cycle. This is diagram of cell cycle. So moving on to the cell cycle. What is cell cycle? It is the life process in which the one cell of an individual is divided into another cell, whether it is prokaryotic, eukaryotic, multicellular or unicellular organism. The life of eukaryotic cell, the cell grows, make new copies and divide. It happens all the time in body, and the division in the body cells is the process in which same sort of DNA is duplicated or copied. So but in the division in the reproductive cells occurs somehow in a different mechanism. We will discuss in another video. What is the difference between diploid and haploid? When there are two copies of chromosome, the cell is diploid, and when the single copy of chromosome, the cell is the haploid. Okay, so the process of phases of cell cycle, there are major two phases of cell cycle: interphase and mitotic or M phase. Interphase occurs most of the time. In mitotic phase, there are four other phases: prophase, metaphase, anaphase, telophase, and then leads to the cytokinesis, in which cell is divided into two. We have a hack to memorize IP on the mat. See, IP for interphase, metaphase, anaphase, I for interphase, then prophase, metaphase, anaphase, telophase, and C for cytokinesis. So what is interphase? It is the largest phase in which 95% of the growth occurs. This is the time between cell division. The cell is growing, DNA is copied and duplicated, and it is prepared for division. The process in which DNA is copied is called synthesis or replication. There are three major phases in the interphase: G1, S, and G2 phases. In the G1 phase, the growth of cell in the size and development occur. We call it differentiation. So the cell is told what to become, what is the fate of the cell. In second phase synthesis, the DNA is replicated, the multiple copies of DNA are prepared. There are three sub-steps of replication or synthesis. DNA double helix is unwinded or separated into single strands by enzyme called helicase. One DNA nucleotide is replicated at a time on the both sides of the DNA strand. For example, A to T and C to G is moved so that two copies of the DNA is prepared with the help of enzyme called DNA polymerase, and the third is the nucleus is left with two exact copies of all the chromosomes. In G2, the cell prepared for division and checks for the error. So it is the preparatory phase for mitosis. This is the diagram: G1, S, G2. We can see that the cell is prepared, and the multiple copy of cells are prepared, and then it is duplicated with duplicated chromosome and ready for mitosis. In mitosis, two duplicated chromosome moves apart, and cytokinesis, it is divided into two daughter cells. So stages of cell cycle I, P, and the METC. Mitosis is after interphase. It is considered cell division. 4 phases: prophase, meta, and telophase. Cytogenesis is after mitosis. So, first of all, mitosis occurs, then cytogenesis, then again mitosis. This is the cycle which continues. Prophase, the first phase in mitosis. There are 3 things to remember in prophase. Chromosome can be seen as 2 chromatid. We can in the shape of an X. Nuclear envelope dissolved. Centrioles are present with spindle fiber. Okay. And next phase is metaphase. Second phase of mitosis. There are also 3 things to look up. Chromosome lie up in the middle equatorial plates. Nuclear envelope is gone. No nucleus. So spindle fibers are on the opposite pole, so that they can stretch the chromatids of the chromosome apart. Okay. Anaphase. Third phase of mitosis. 3 things to look. Spindle fiber pull the chromosomes towards the separate pole. Chromosome must split in half. Sister chromata are now their own chromosome. The telophase, the final stage of mitosis. 3 things to look. The nuclear envelope again reform. So each set of chromosome are that we can now call up daughter cells. They have each set of chromosome. They can uncoil. Spindle fiber are moved or gone. Cleavage is forming between the cells, so that cell can be separated into 2 daughter cells. Cytokinesis. After that, the mitosis cytokinesis occurs. Its final step in cell cycle. It means the movement of daughter cells from each other, the final pinching of cell into the 2 completely identical cells. This is the diagrammatic explanation of whole cell cycle. After G2 phases, in the G2 phase, nuclear membrane, sister chromatid are present. You can see that in the diagram. Centromere is present, and replicated chromosome, they are loosely coiled. What happened in prophase? The nuclear membrane. This is the nuclear membrane. This is finished or moved in this step. Spindle fiber appears, chromosome condensed. Okay, next is the prometaphase. The spindle fiber they attach to the chromosome condenses. In metaphase, chromosome align on the equatorial plates. In anaphase, centromere divide and sister chromatid move to the opposite pole. After that, the telophase, nuclear membrane reform, spindle fiber disappear, chromosome decondense, and cytokinesis, cytoplasm divide. Cytokinesis is the division of cytoplasm. Parent cell become 2 daughter cell with the identical genetic information. Remember, in mitosis, the genetic information is exactly the same copy of our parent cell. So the photocopies of the parent cell are formed. Certain gene enzymes trigger the start of cell cycle. Replicant also tells the cell what to do. An important cell cycle in the unicellular, they reproduce by cell division. Multicellular, they help in growth and also help in cell repair. Okay. Cell cycle, multicellular growth, increasing number of cells and size of cells. Interphase G1, differentiation, cell told by gene to become specialized cell. For example, cell told to do that job. Morphogenesis is the pattern formation of specialized cell to become tissue. So they help to form specialized tissue. Disruption cell cycle, a certain enzyme gene tells the cell cycle to begin to rapidly cell division become out of control. This is called cancer. This is the development of multicellular organism, animal and zygote to 8 cells, blastula to gastrula, okay, to the adult star. This is plant and zygote 2 cell, octant, heart stage embryo and then plant.